There are different types of PCR: reverse transcriptase PCR, real-time PCR, nested PCR, step-down PCR, to name a few. We will see how to perform a standard PCR. As our first step, we isolated the genomic DNA from bacterial cells. After the isolation of genomic DNA, we next amplify the gene of interest using polymerase chain reaction. Materials required are template DNA, tag DNA polymerase, forward and reverse primers, deoxyribonucleotides, tag buffer, nuclease-free water, thermal cycler, micropipette tips, micropipettes, and mini cooler. The concentration and volume of reagents used for a 25 microliter reaction mix are given as below. Pipe the laminar air flow using 70% ethanol. Place the accessories as shown. Now, add 2.5 microliter of 10x buffer. Buffer is added to maintain the pH. Standard PCR buffer contains 50 millimolar potassium chloride and works well for amplification of segments of DNA greater than 500 base pairs in length. Raising the potassium chloride concentration to 7200 millimolar often improves the yield of shorter DNA segments. Now add 0.5 microliter of 10 millimolar DNTPs. Standard reactions contain equimolar amounts of DATP, DTTP, DCTP, and DGTP. Add 1 microliter of 10,000 picomoles per milliliter forward primer. Now add 1 microliter of 10,000 picomoles per milliliter reverse primer. A pair of synthetic oligonucleotides are added to prime the DNA synthesis. This quantity is enough for at least 30 cycles of amplification. Higher concentration of primers favor mispriming, which may lead to non-specific amplification. Add 0.2 microliters of tag DNA polymerase. The concentration of tag DNA polymerase is 5 units per microliter. Now add 5 microliters of template DNA of the concentration 2 microgram per milliliter. Add 14.8 microliter nuclease-free water. This makes the reaction mix to 25 microliter. This is a thermal cycler. Switch on the thermal cycler. Place the PCR tubes in the thermal cycler and set the thermal cycler for the following conditions. Initial denaturation at 95 degrees centigrade for 5 minutes. Step 2, denaturation 95 degrees centigrade for 1 minute. Annealing at 55 degrees centigrade for 1 minute. Extension 72 degrees centigrade for 30 seconds. Go to step 2 and repeat the cycles for 30 times. Final extension 72 degrees centigrade for 2 minutes. In some cases, 1/5 volumes of mineral oil is added to prevent the evaporation of reaction mix. But this poses a problem while loading the sample for gel electrophoresis. we use a thermal cycler of heat bonnet type that prevents evaporation loss without the use of mineral oil. As you can see, cycle 1 has started. It takes some time to reach 95 degrees centigrade. Once 95 degrees centigrade is reached, the reaction starts. Initial denaturation occurs where the two strands of DNA break apart. When the initial denaturation step gets over, it goes to the next step. Where it cools to 55 degrees centigrade for annealing. During annealing, the primers go and bind to the gene of interest. After 30 seconds of annealing, we have an extension step at 72 degrees centigrade for 30 seconds. During the extension, the Taq DNA polymerase binds and extends the primer, thereby creating new strands. Now, as you can see, when the cycle is over, it gets to the second cycle. The steps from step 2 to step 4 repeats for 30 cycles. Thus you can see an exponential rise in the number of fragments. After PCR, analyze the sample using agarose gel electrophoresis.